Davis, and her research focuses on PRRS evolution, PRRS classification, molecular epidemiology, and the dynamics of pathogen spread through animal populations. Her lab has been instrumental in the adoption of lineage and variant-based nomenclature for describing the genetic diversity of PRRS in the US and how that diversity has shifted across time. So I think, Kim, you're going to be the last speaker of the morning, and maybe we're going to see some sort of summary from all the presentations. Maybe it's putting a lot on you, but we're looking forward to this provocative title that you have in your talk.
All right, thank you so much, and to Maria and Mark for inviting me and for putting together this session. So this talk is really supposed to be a synthesis about how we think about PRRS epidemic waves, and it's probably just not any one factor that drives the dynamics we see in this country, but how multiple factors are actually converging to really give rise to the new epidemic waves that we see. I'm going to start by acknowledging that a lot of the work that I'm presenting here is either the products of my lab members as well as other collaborators at the University of Minnesota. I'm kind of trying to be the torch bearer for a lot of the members of the Swine group at the U of M, so it's not all my work. All right. So before we get into the question, is PRRS worse now than it used to be, we're going to just take a step into, well, how do we even classify new versus old strains? So at least we have a kind of common place to talk about new and modern PRRS. We'll today be using the lineage and variant classification system, which just briefly to orient you, this is a classification system based on phylogenetic relationships and of different viruses based on their OR5 gene. And a lineage or a sublineage basically attempts to group viruses according to their kind of ancestral family group. And typically we see about a 9% or greater difference between viruses but belonging to different sublineages. Now, these sublineages are still really big and quite diverse. So in the past few years, we have further stratified these sublineages into smaller genetic groups that we have referred to as a genetic variant. So now here in the small box, you see lineage 1H, and then the colors within that box are different variants that exist within that sublineage 1H. Typically, viruses belonging to the same variant have an, on average, greater than 97% similarity on their OR5 gene. And so anytime that you see just a number followed by a letter, like here 1H and 1C, that refers to the sublineage. And then if there's a decimal point followed by an integer, that's a variant designator. So here we have 1H5 and 1C5. So these are the fifth variants that have been identified in those respective sublineages. Okay, so once we are able to classify our viruses, we can start to look at kind of the macro epidemiological dynamics through time. This is a plot up here showing the effective population size of different sublineages within lineage 1 over the past, say looks like the past 25 years. And the thing that I want to draw your attention to is that every few years we get the emergence of a new sublineage. And that's followed by a period of exponential spread, and that the steepness of that population expansion of that variant is pretty consistent, or that lineage is consistent across time, right? And so these are the epidemic waves that I'm really talking about when I say that we experience these waves in the country. And then eventually that sublineage will peak and then begin to decline as something new takes over. And we can actually extrapolate the timing of those peaks, and that's what the stars represent in this timeline. And we see this really consistent time. This is probably the PRRS clock, that going all the way back to 1998, we see on average, or actually this is a median, every 3 years we see a different dominant sublineage in the country. Now, more recently, we have started to use the variant level nomenclature, so really starting applying this to data from 2020 onwards. And so here is this figure from up to 2019 and the lineages on the left, and then on the right we can start seeing the dynamics of our 1C5 emergence event, which follows, again, a very similar steepness of that curve and how fast it was expanding. And then in the last year, we have the 1C532s in the Midwest. Again, the steepness of that wave is very similar. These 1C532s, this is an offshoot of the 1C5 variant that is genetically diverged from the 1C5s. So when I talk about old versus new then, I would say that our historic lineages are our lineage 5s and our lineage 9s. Those peaked 1998, 2001. If you're seeing, basically we do not see any wild type virus left, at least in the US, that belongs to lineage 5 or lineage 9. So I'll call those our historic lineages. Then in the early 2000s, we have our early lineage 1 viruses. So the MN184 isolate belongs to lineage 1F. And so these we'll call the early lineage 1s. And then our contemporary lineages, I would call the 1As, which includes our RFLP 174s, the 1Hs, and now we have our 1C5s and its offshoots. Okay. The other thing I want to establish is really a discussion about what do we mean are recent variants more severe than we saw historically. And severity is a complex term, and different people mean different things when they say, is it more severe? So we could define severity as virulence, and this is a term that is often misused. But on a virological standpoint, the definition of virulent is the harm caused to the host by infection with the virus. We have different ways we can measure this. We can measure this in cells, looking at cytopathic effects in an in vitro type assay. We could measure this in pigs, looking at mortality, reduced growth, fever, clinical signs, all those things. Immunoreactivity, we had to talk about that from Dr. Rahe. Or we can measure this in farms, looking at percent mortality, percent abortions, et cetera. So those are all different proxies we can use to get at virulence. Now, commonly in the industry, when I hear people talk about severity, they really are talking about high fitness viruses. Higher fitness fitness is defined as the ability of the pathogen to replicate and spread rapidly. So it's more of an epidemiological term. And when I hear people talk about this as a bad PRRS, they often mean it's spreading really fast. We're seeing it crop up everywhere in a short period of time. And so this is another definition of it's a bad PRRS, right? And there's different levels or different scales we can measure that. We can measure the fitness of a particular virus in vitro. We call this replication fitness, which is basically how fast can that virus replicate. We could measure this at the pig level, looking at transmission fitness, how efficiently is it spread between pigs. So we might look at things like shedding or the transmissibility. But we can also ramp that up and look at population level, all the way up to the regional and national scale, where we're looking at observed prevalence and observed incidence. And that's where we really start to say, well, it's really prevalent. The incidence is increasing really rapidly. This is a bad variant of PRRS. And a lot of these things are correlated with each other as well. All right. So a lot of different ways we can think about the severity of PRRS. But in total, we probably have some concept that a bad PRRS is a combination of virulence, so the harm caused to the individual infected pig, and something about fitness, so how transmissible it is and how rapidly it's spreading, and those things combine to what we kind of perceive is PRRS severity. The other thing that we'll note too is that there's a number of things that affect this. For example, the immunity in the pigs or the population is going to affect transmissibility. It's going to affect virulence, so that plays into this. Immunity and viral evolution are going to expect it to interact with each other, and that's going to determine the fitness or the ability of different variants to spread. And so the first part of my talk will focus on the evolutionary perspective of PRRS severity and how that might interact with immunity. Well, let's take a shallow dive, we'll say actually, into the immunological perspective. And then part 3 will look at the transmission perspective of PRRS severity. Now, in the course of this symposium, we've also talked about farm management. That's going to affect immunity. That might directly affect severity as well. It's going to interact with population factors, co-infections, all of that plays in, and all of that's related to, underpinned by pathology in the animals. And I'm not going to touch a lot on those because we had some great talks on that. And all of these things combine, really to determine production impact, that we feel from different variants of PRRS. And that's probably ultimately what we care about when we're saying that this is a severe variant of the virus. Okay, so let's start. Number one, evolutionary perspectives of PRRS severity. I study viral evolution, and I get this question a lot. Do new lineages evolve faster? Is the virus evolving quicker now than it did historically? And we know that even compared to other RNA viruses, the rate at which PRRS type 2 changes is exceptionally fast. However, when we estimate that evolutionary rate for different lineages, or sublineages in this case, of the virus, we actually don't really see much of a difference as the newer sublineages accrue mutations in their genome at about the same rate as the older sublineages. So it doesn't seem that it's evolving any more rapidly now than it used to. Now, the accrual of point mutations is just one mechanism of evolution. The other one is recombination. And recombination is, just to refresh your memory, when a pig or a cell is co-infected by two different variants of the virus. As those viruses replicate in the cell, they can exchange some of their genetic material, and what results is some sort of progeny virus that is a mosaic genetically of the two parent viruses. All right, so has recombination become more common? And my answer to this is maybe. And a few caveats on why this is maybe. First, are we better able to detect recombinant viruses now? We have a lot more whole genomes available to us, so probably we have more data. And we also have more overall genetic diversity now, so we see recombination events with different lineages as the two parental viruses, so we're able to detect it easier. It's a bigger genetic change when those viruses recombine. My other comments on recombination as it more common is what do we mean by recombination rate? Do we simply mean the prevalence of recombinant viruses? So of all the genomes we have, how many of them are recombinant? And that's one way to look at it. This is this paper here. I'm showing data from a group in China. But they showed that interlineage recombination prevalence is higher, 62% of sequences in the US than in China, where they have 28% of sequences. So we can look at this and say, okay, recombination is common. But there's a difference between looking at just the prevalence of recombinants and then how often recombination is actually occurring. So I'll give you an example of what I mean. Frequency of new recombination events. 1C5, that variant of the virus, those are all recombinant viruses. We did the analysis in 2021 with I think somewhere around 20 whole genomes from 1C5. And at that time, all of those viruses had the exact same recombination profile. They all descended from the same basic recombinant ancestor. But there's thousands of those sequences in our database because belonging to 1C5. So how do we count that? Is that one event, one recombination event that has happened, and we should be counting that? Or should we count it as the prevalence of all of those descendants of that event? So this is why it's really hard to say, is recombination more common or not? Nonetheless, if we look at these plots in China, this is time in the bottom. The blue is our recombinant genomes, and the green is non-recombinant. And yeah, in China, it looks like the blue bars are inching up. They're getting higher through time. So maybe it's getting more common in China. In the US, if you squint at these small bars here, we don't have a lot of whole genomes from back then, but the height of the blue and the orange bars relative to each other is about the same through time. So I don't think in the US we actually have a lot of evidence that recombinant viruses are being detected at higher frequencies. To me, this looks like it's about the same through time. So maybe recombination is more common. Nonetheless, is PRRS more genetically diverse today? Absolutely, it is more diverse. This is a plot of how lineage 1 has diversified through time, and you can just see this radiation of genetic diversity color-coded by all the different sublineages we have for that virus. So it is more diverse, and as it evolves genetically and diversifies genetically, that does mean that it probably is changing antigenically, and evolution doesn't occur in a vacuum. There's been a lot of theories thrown out that PRRS evolves to evade immunity. So if you have an immune population, that is going to favor viruses that are evolving to evade that immunity in the population. We just published a study in July on this topic because it's been thrown out there a lot, but the causality of immunity to evolutionary change has never really been shown in a controlled experimental setting for PRRS. So in this study, which we had a number of contributions in this, from different labs in this room, I'll highlight Declan Schroeder and Maxim Churin were instrumental in this project and their labs. But in this study, we set it up with 3 treatment groups. We had a group of non-immunized pigs. And then 2 different treatments of vaccinated pigs. One group was vaccinated with Prevacent, the other one with the Engelvac vaccine. Those pigs were kept for 64 days post-vaccination, and then we challenged them with a lineage 1A 174 virus, and then let that virus be in those pigs for 14 days. At the end of this 14-day period, we took whatever viruses were in those pigs, and we inoculated that into a next batch of pigs, keeping those same treatment groups. So we did this across 6 batches of pigs to try to simulate an infection chain of pigs, and then allowing the virus to evolve across those immunized or non-immunized infection chains. In total, we were able to recover 110 genomes, whole genomes from this experiment from 127 pigs across this 84-day chain of infection. And I'm going to, the paper's got a lot of results in it. I'm going to distill it down all into one single figure, and that is that the more genetic change was accrued in the vaccinated pigs than in the non-immunized pigs. And that's really what this figure shows. Here we have batch 1 through 6. The green line at the bottom here is the evolution of that challenge virus in our non-immunized, so our non-vaccinated pigs. And you can see that the line stays pretty close to 0. And the y-axis here is the distance, the genetic distance to the challenge virus. So it stays pretty close to the original virus in our non-vaccinated pigs. And in both vaccinated groups, we see this kind of drift away or movement away from the original challenge virus in terms of genetic distance. So it does seem that that immunity in our population does, we'll say, accelerate genetic change in the virus. Now, the idea of why this would work or why this would happen, of course, is that the virus is evolving to evade host immunity. And that really brings to the second portion of this talk on the immunological perspective of PRRS severity. I am not an immunologist, so I really appreciate the talk that Mike gave before me. But I will give you our take, or some of our take, and some of the research we're doing in this area. So the question here then is, how does genetic change impact immune recognition of the virus by the host? First, we'll talk about antibody-mediated immunity, and for this portion, we're going to really be focused on the ectodomain of the virion, because that is the part of the virus that is exposed to antibodies, and that's where the antibodies would interact. So we have done some work on cross-neutralization, really where we generated antisera in pigs to a diverse set of PRRS isolates. And then we then did a cross-neutralization panel with, again, a diverse set of PRRS isolates to really quantify the ability of antisera generated against isolate A to neutralize or recognize and neutralize isolate B. So in this plot here, each of these dots represents that kind of pairwise neutralization that was done between virus A and virus B on a cross-neutralization panel. And we have plotted on the x-axis is the OR5 genetic distance, and on the y-axis is a normalized neutralization titer, essentially. And so if the value is 0, then that's kind of average neutralization. If it's above 0, it's above average neutralization, and then below 0 is less than average neutralization. And the first thing to notice here is that there's maybe a general correlation between genetic distance and normalized cross-neutralization titers. There's a lot of noise here in the data. A few interesting observations for me is that we have, if the viruses are within 2.5% of each other, all of the points cluster in this above average neutralization area. And if it's a greater than 7.5% genetic distance between those viruses, all of the data points cluster around the average neutralization. Now, we don't have any data points in between because we did not have any pairs of viruses in our assays that were more around the 5% range. And that 5% range is often what we might see if we're dealing with co-circulation of viruses in the field. So we'd really like to fill that in. We see a similar picture if we look at specifically the ectodomain amino acid distance rather than genetic distance. There's a correlation. There might be a fall off maybe past 2.5%. But there's a lot of spread in these points, right? There's a lot of noise in the data. And so what we have tried to do is go beyond just looking at genetic distance, which is essentially just a percentage of sites that are different between two viruses, and actually say, well, not all sites are equally important, right? If we just look at a percentage, we're saying any change is the same, and that's probably not true. Some changes maybe aren't that meaningful, and others might be very meaningful. So we have applied machine learning algorithms to try to predict this data better. And we found that we were able to predict above or below average neutralization from the sequence data with around 88% accuracy with our internal data. That's from our own project. And the key predictors that were important in making these predictions was the amino acid distance, but also changes at particular amino acid sites, right? These sites that are in epitopes, and that helps us improve our ability to predict this. We also applied our model to external data because we wanted to have some external validity. So we applied our trained model to virus neutralization titers coming from other studies, and this study that we used was actually an NCSU study, as well to lung lesion outcomes from challenge studies. And we found that we were able to predict with greater than 75% accuracy on that external data, which we're excited about. And of course, there's a lot of caveats to this data that my postdoc is going to present on more details in the AI session on Tuesday, Dr. Poranjanavikun. So if you want to know more about the limitations of the data, you should go to his talk. But we have put up a web tool that allows you to upload your own sequences and get this sort of estimate with the caveats that we try to outline very specifically here so that you can do this sort of quick bioinformatic estimate with your own data. Now, one of the limitations of this is all neutralization data based on antibody-mediated immunity, which misses the whole component of what PRRS does in a pig on an immunological perspective, which is cell-mediated immunity, right? And so in a different study, we have been focusing more on cell-mediated immunity from a bioinformatics standpoint. Just to remind you briefly, I always like to refresh myself because I'm not an immunologist, but cell-mediated immunity essentially happens when the virus is ingested into an antigen-presenting cell. In that cell, I'm probably making it very simple, but the viral proteins are chopped up into little pieces, and then those pieces are presented on the outside of the cell. And then those short viral peptides or little pieces of protein may be recognized by a T cell, but that's going to be based on the genetic sequence of the amino acid sequence that's present in that short peptide, as well as does it bind well with the T cell, the SLA. All right, so we've been working with a company called EpiVax, which has is a bioinformatic platform to essentially estimate with computational tools where these T cell epitopes are in the viral genome. And essentially what we've done is we've analyzed the structural proteins of over 150 PRRS whole genomes. And then we can, from there, calculate the percent of shared T cell epitopes between each pair of genomes. So what I will point out here, and just to orient you, each of these dots represents a genome we assessed, and the closer these data points are to each other means that they had a higher percent shared T cell epitopes between those two genomes, okay? So that's what this plot shows, and then to walk you through that, there's 3 main clusters that we can identify in this kind of shared T cell landscape. We have the green cluster, and this cluster includes our historic lineages, so the lineage 4s, 5s, 6s, 7s, 8s, 9s, and our early lineage 1s, so the 1Fs and the 1Ds. They all belong there, and those diamonds, those are all of our modified live vaccines that are on the market. All of them live in that green cluster. Now, the blue cluster and the red cluster are our current prevalent lineages, so the 1Cs and the 1As and the 1Hs, all are up in those 2 clusters, right? So not only do we seen a big shift in our T cell landscape away from the historic viruses, but we also have now 2 clusters that exist in our US population, and they are different each other, at least computationally. This needs to be validated in a lab, but it means that we have a lot more diversity out there than we used to from a T cell perspective, or a T cell epitope perspective, and that we don't see good overlap with any of our vaccines, okay? So, we know that the virus is evolving. We see genetic diversity, antigenic diversity, both from a T cell perspective and a antibody, or a humoral immunity expected. What is the implications of this? Well, one, we talked about recombination earlier. Well, now because we have so much diversity, when recombination happens, it can be with viruses that are really genetically and maybe antigenically distinct from each other. So we have the potential for these recombinant viruses to now have more drastic shifts in their phenotypes, right, in their behavior, because you're mixing things that are more different from each other than they used to be. Other implications, we pretty much live in a world where all immunity is heterologous, right? Unless you're maybe doing LVI in your specific farm, but if you're looking at more of a regional or circulation level, all immunity is heterologous. This, of course, affects our efficacy of our immune interventions, but also it means that if my farm went through an outbreak 6 months ago with one variant of the virus, well, this diversity means that there's a lot of viruses circulating probably in your area that are antigenically different and are still probably going to be able to successfully infect those pigs because we have that enormous genetic diversity out there, right? So these things are going to combine to make the virus harder to control probably than it used to be, okay? All right, part 3. The transmissibility perspective of PRRS severity. Has the transmissibility of PRRS changed? Okay, let's talk about transmission. For successful transmission to happen, it requires a pig that's infected, and that virus is replicating in that pig, and the virus is being shed from that pig. Then there has to be a susceptible host, and a sufficient amount of virus has to reach that pig so they can get an infectious dose of the virus, and then it will become infected too. And then that's going to be modulated, how much dose is going to reach that susceptible pig. It's going to be modulated by transmission routes and survivability and other factors that could affect that. For this portion of the talk, I'm going to highlight 2 studies that were experimental studies on kind of variant level or strain level variation in the first one, airborne transmission. And that study, I'm actually not involved in that study. It was led by Dr. Juan Mena and Monse Tormorel. And then the second study is Melini et al. 2025 on infectious dose in shedding. And Marcelo is a PhD candidate advised by Cesar Corzo. And both of these studies used the same 3 variants or isolates in their experiment. They use A1C5, which we've heard a lot about today. They've used lineage 1A174, which is fairly new, but we'll say it's getting to be 10 years old or 12 years old. but I would still call it as part of like the newer viruses that we see, and then a historic strain belonging to lineage 9. Okay, so same 3 sets. We'll go to the airborne study first. In general, and this is seen in the numerous studies that are published now and some data that's been presented by other groups today, is that the newer viruses, the 1A and the 1C, had worse clinical outcomes than the historic lineage 9 virus. So that's shown here in average weight gain, but it's been shown in many other studies as well. Also, this study showed that the nasal shedding of all 3 of these viruses was relatively similar. And this is also consistent with what has been reported by other studies, that you don't necessarily see a lot of differences in nasal shedding. Now, what they did find a difference is the ability to detect the different viruses in air through time. Here we're talking about up to about day 19 of the study. And it seems that the lineage 1A viruses had a more sustained detection in air compared to both other viruses. The 1C5s actually were probably the ones that had the most drastic and notable decrease in their airborne detection through time, whether we look at air samples or surface deposition, where we really see a strong drop-off after day 9, day 10, post-inoculation of these animals. And then lineage 9, this historic strain, was somewhere in the middle, okay? So maybe there's a difference in this kind of transmission route. I think that there's a lot more research to be done in this area. The second study on infectious dose and shedding. We briefly talked about this one as part of Mike's talk, but the study design essentially had 3 different isolates belonging to 1A, lineage 9, and the 1C5 variant. And then there were pigs that were inoculated with different concentrations, so a different infectious dose, starting with a 10 to the 4, and then that was diluted. There was pigs that got a 10 to the 3 dose, 10 to the 2, all the way down to 10 to the 0. And then those pigs were followed for 30 days. So all of the pigs, when you inoculate them with 10 to the 3 or 10 to the 4, which is a fairly typical dose we'd give in an experimental study for PRRS, pretty much all the pigs were infected by day 4 post-inoculation if you give them that sort of dose, regardless of what variants you give them, okay? Now, where the differences really emerge is when we look at these really low doses, where the lineage 9s and the 1As, you don't see much going on at day 4. 0, 1, maybe 2 pigs out of 6 infected by day 4 at these very low doses. The 1C5s, by day 4, you have 4 out of 6 or 5 out of 6 pigs infected or positive at day 4 with those very low doses. And if you extrapolate out the math, which is done in the paper, is that what you get at is that the infectious dose for 1C5 is about an order of magnitude less than these other viruses, right? So what's the implications of that? Well, this has implications for biosecurity. If the virus can infect at a lower infectious dose, it may be able to maintain that dose through the biosecurity process. It's going to make your biosecurity appear leakier, right? You have to be much more effective at killing all the virus or preventing more virus, get that virus down to a much lower level to be able to properly protect susceptible pigs, right? So it's going to make it seem leakier, or it's going to make you think maybe it's transmitted in a different way because our biosecurity is not working anymore. But this could be related to this really low infectious dose. It's going to be leaky biosecurity. The other implications of this is rapid between animal transmission, right? This data comes from a study, VanderWaal et al., 2023. This is an ISU study. But essentially, they have data on this. They had a number of different isolates belonging to different variants that they inoculated to animals. Then they introduced contact animals into those pens and saw how quickly those contact animals became infected. And at day 2, post-contact, so post-introduction of these naive animals into these pens, you can see the 1C5 variant, 4 of those animals were already infected. And that's not true for any of the other viruses that were used in this trial. Most of those other viruses took to day 5 post-challenge, where you start seeing the majority or all of the animals infected. So there's a difference there. I took this data and I did some back of napkin math, and don't quote me on this because this is a small sample size, and I did this in probably a fairly rudimentary way, but for all contact pigs to be infected by day 2, like the 1C5, you would need an R0 of 5.5, which is on the upper range of what's been estimated in other studies for PRRS. Now, if you change that to day 5, for all the contact pigs to be infected, that R0 goes down to 2.25, so about half is transmissible, or 2.25 is probably more like typical PRRS anyways, so that would make the 1C5 variant about twice as infectious, or transmissible, we'll say. I then plugged that into a simplistic epidemiological model. This is a SIR model where I kept everything the same and I only changed the R0 just to get an idea of what does this look like in a population. What's a R0 of 5.5 look like compared to a 2.25? So here we have time after introduction. Here we have the percent of animals infected. And this curve here is the 1C5-like scenario with the R0 of 5.5. You get a really spiky, rapid outbreak that explodes compared to an R0 of 2.25, which is our more typical PRRS. So even if you have ultimately the same number of animals infected by the end of your outbreak, and even if the per animal outcome, per animal chance probability of mortality is the same between these two scenarios, I would bet that that's going to feel a lot worse, right? This is going to feel horrible because all of that mortality is happening in this really short period. Your opportunity to intervene is super short. That's going to probably feel more severe than this, right? I think we'd all agree on that. And so my point is that even if your per individual outcomes are similar, maybe your pathology is similar, this timeline is going to make it feel worse, right? So we're really getting at transmissibility matters in terms of our perception of PRRS severity, and the 1C5 seem to have this really, really transmissible phenotype. Okay, so last part, I'm going to share some data that was shared with me by a new collaborator that we've been working with, Dr. Federico Zuckerman at University of Illinois, who shared this slide with me to really get at, well, okay, are some viruses just fundamentally more fit? And in this study, he has an assay, an in vitro cell replication assay, and he's been able to test a variety of PRRS isolates where he knows in the field what that virus did in a sow population. And he's classified the viruses into either these sort of high mortality viruses that cause high mortality both to the sows and pre-weaning mortality, as well as then the more conventional, typical PRRS mortality isolate. So sow mortality less than 1% for the conventional, more than 10% for high mortality. When you put those isolates onto cell lines and do a one-step growth curve, we can see that these high mortality isolates are really good at replicating in cells. This was highlighted by Dr. Rahe about the ability of virus to replicate, and really showing that these high mortality viruses replicate more rapidly, and then they have a higher overall burst size, which really emphasizes that these viruses are really fit. They replicate well, and that seems to be related to clinical impact. I also believe that the lineage 1 viruses just in general are just more fit, inherently more fit than our more historic lineages. And I'll give you two kind of more epidemiological data points on this. This is on the left, the US, and you can see that from about 2009 onwards, lineage 1 and its sublineages has taken over. Lineage 9 used to be far more prevalent before that period. Lineage 1 most likely came from Canada and has completely displaced everything else that was there. Now, that's just one data point, really, right? That's just the US as one data point. But the exact same thing has been happening in China. Historically, they've had lineage 8 viruses. The lineage 8s include the so-called high-path PRRS that we heard a little bit about today. It's a lineage 8 virus. That used to be the predominant virus or lineage in China. And then lineage 1C was introduced, and then more recently 1A was introduced. Here, both are just represented in the red color. And you can see that lineage 1 is taking over and kind of displacing the lineage 8s. So it does seem that this virus is just really good at outcompeting the other viruses. Okay, so closing thoughts here. We have described how we have these recurring epidemic waves in the US driven by the continual genetic evolution of the virus. But what really sets the conditions for our perfect storm where we have one of these explosive spreading events? Well, we have evolutionary change. So we're always generating viruses that are just a little bit genetically different and probably different antigenically too. So we have that kind of pool of viruses that one of those could be the next big one, right? We also live in a world of low population immunity. The majority of our population probably has some immunity to PRRS because they've either seen it as a natural infection or they've been vaccinated or immunized in some way. But we know that there's poor cross protection, and so we're never going to really achieve good herd immunity. And we also know that immunity may drive the genetic shifts in the virus population through time. So we're laying the groundwork of a susceptible population with the right antigenically different virus can sweep through it, okay? We also know that our newer variants and our newer lineages appear to have faster replication, which could be linked to virulence, and they appear to have higher transmissibility, perhaps related to infectious dose. And these two processes of how the virus is becoming more fit on a transmissibility standpoint, maybe fundamentally more fit, with this issue with low population immunity from a cross-protection standpoint really set the stage for this. Now, not all variants are created equal. Evolution produces both winners and losers, right? So at any given point in time, including today, there's a lot of genetic variants out there. Some of them probably are winners. They're going to spread, or maybe that's losers for us, but, and some of them are less fit. This is a time-snapped tree every year for 5 years, and you can see that some variants, you know, they explode. This is the 1C5 variant. It just explodes. And then there's other variants that they just kind of sit there and they don't do much. So we've been working on how can we actually anticipate which variants are going to be the next big one. We have an initial paper out on this led by my postdoc, Dr. Parmar Chanavakun. We have a version 2 of this as well that we think we can use all of our retrospective data on these kind of trajectories of different variants and build a model that predicts, well, which variants are likely to grow by more than 20% in the next 12 months. And we have a model that we think works pretty well to do that. We've taken that one step further. This is a project led by Dr. Mariana Kikuchi that takes that information and then cross-tabulates back to our site-level incidence data in MSHIMP, and then produces a monthly report on variants under monitoring. So we identify the ones that we think that have concerning trajectories, and we start putting together the epidemiological information and producing reports. This report has already been being put out for the last, I think, few months now. But essentially, we try to make this information available to industry, so if they see this variant, they have the situational awareness to know is this part of one that we think is particularly concerning. And the idea here is that we're trying to get ahead of the curve a little bit on some of these emergence events, rather than identifying them already when the forest is on fire, right? And we really think that this is the first step. If we can start getting better communication about it, that's the first step to a better response as well. So that's the kind of where we're going with this. I'm going to leave it there so that we can have some questions or discussions. So thank you all for your attention.
Thank you very much, Kim. Do we have questions right away from the audience? I was going to ask him, from what you showed and what we're seeing, do you think it will be maybe beneficial from the diagnostic point of view, or I don't know, maybe clinically? We take the hit really hard at first with these new variants, but then that will be beneficial later on because there will be like less lingering of those variants, or would that be I don't know, something that is not going to be observed. So it will be bad all the way through.
Yeah, I mean, I think looking at the longer timelines, and I was noticing this about the data that Danielle presented, is that, and this is true, I think that from the production data he presented on the lineage 1As, which include our 174s, but it also seems to be true for the 1C5s, is that the clinical impact seemed more severe early in those emergence periods and then has kind of attenuated through time. And that is something that we would predict with viral evolution, but also could have to do with, once a large proportion of the pigs have seen that particular variant of the virus, there might be a lot of immunity towards that, and that might be what's driving that. But it does seem that the viruses seem to be more severe in that early part of the emergence period.
Thank you, Kim. That's like quite very impressive presentation. I have a question about the chart that you have. In one of the neutralizing antibody and between the genetic distance. So the genetic distance 0, you always have higher like neutralizing antibody, like above the average, right?
Mm-hmm.
So it, please correct me if I'm wrong. So does it mean that the homologous like strain will confer like higher, much higher like neutralizing antibody to the current strain? So to step further, does it mean that autogenous killed vaccine from the field will provide better immunity?
Yeah, okay, so that's a great question. When you have the genetic distance of 0, the way that study was done is that we did have the virus neutralization titers from the antisera to the exact same virus that was used to generate the antisera, so it's true homologous, and you would expect that to be higher. Interestingly, we saw though that up to around 2.5% still seemed to be equally as high, so it seems to be like some level of homology or homologous recognition at that level. And we do have whole genome data for that, so it's not just OR5. But the question about killed vaccines. Maybe, I mean, in theory, yes, but it's a killed vaccine, so you're not going to get the same sort of response. There is a talk in the PRRS current events section later, I think it's tomorrow, that talks a little bit about how we might be able to boost immunity with killed vaccines in a study that's not published yet that addresses that. And so I encourage you to go there to address that fully. It's good to get that good match, but it's a killed vaccine with limitations of that. So, yes.
Kim, I have 2 questions. I'm sorry if I asked something that you mentioned and I didn't get. Most of the sequences that we have are from the sow herd, but the sow population is replaced at 60% rate a year, and then in a fast-evolving virus, the growing pig is replaced at a 220% rate a year. So what are your thoughts on, are we missing most of the evolution because we're just sampling the sow, most of the sow population and not the growing population, which is where the virus, the majority of the population is?
Yeah, I mean, I think that there's more sequencing being done in the grow finish than we might think that there is. If we look at the number of sequences we have from grow finish, it's not hugely disproportionate on how much sequencing has been done. And then the other thought of that is how much of those sequences that might be unique in the grow finish are going to eventually find their way to a sow farm anyways, and you would still flag them at least at a kind of regional scale. So I think we're still working on that about where do we see variants emerging. But I do think that the grow finish population is a dangerous population in terms of recombination because you do see a lot of mixed infections in those herds. So their contribution, they're important to that kind of mixing population for mixing viruses is substantial.
Okay. And the other question that I have is, the tools that you use to map or to understand the cell epitopes, have you used those to understand the neutralizing epitopes? Do we know what's the epitope? Like at the complete genome level, do we know where are the variations or where are the neutralizing?
Yeah, so from the T cell epitope perspective, there's some research from NCSU that really starts to cross those things in terms of trying to identify which. There's T cell epitopes everywhere. There's a lot of T cell epitopes in those viruses, and which proteins are most important, bear the most, and the most important for neutralization. It's tricky because T cell epitopes can be regulatory as well, so they can have different roles in the response. But based on kind of reading the literature and some inference, I think that, and from some of the studies we've done, I think that GP5 is not where you want to look for cross-conservation of T cell epitopes as being a meaningful measure of potential cross-protection. GP5 might actually be the least useful one to look at from a T cell perspective. I would point to GP2, M, or the E protein as being potentially more interesting areas to focus on if we really want to look at does conservation or sharing of T cell epitopes translate to cross-recognition of different viruses and cross-protection. Not sure if that got to your question or not.
Kim, it answered mine, so that was good. Over here. So another question on the T cell epitope stuff. So y'all looked at structural proteins. What about the non-structural proteins?
Great question. The grad student that's leading that work, Julia Baker, just spent the first part of September at Epivax, which is the company that has the platform. And while she was there, she ran all the NSPs. And well, we haven't finished analyzing the data yet, so stay tuned.
Thanks, Dr. VanderWaal. Another enlightening talk. You talked about the R naught of those two viruses, one being 5 or close to 5 and the other one 2. And that's the speed at which that virus transmits within a population, correct? And so are the same number of animals being infected just at a different time rate, or is that an indication that those viruses will die out quicker?
So if you do a simple SIR model on that, you would result in the same number of animals that would probably get infected ultimately. But in reality, we're doing interventions along that, so you probably wouldn't see the same number of animals. So it depends on what's actually going on in that population. There is some thought, though, that these highly transmissible viruses should burn through that population. So assuming that you're not introducing susceptible animals to a population, if you have a really high R naught, you're going to burn through those susceptible animals much quicker and should resolve more rapidly. Now, whether we see that in the data, the time to stability, would be interested to look at that. But in reality, our populations aren't truly closed, and we're doing other interventions. So it's not necessarily, you can't extrapolate from a simple model to what would actually happen.
Well, once again, thank you very much, Kim, and thanks to all the speakers this morning.